Thanks, thanks much for the invitation to speak to this group. And I want to talk about water, and as the title suggests, something about a fourth phase of water. Why a fourth phase? Well, you know, to start with, I'll explain that later. To start with, most people presume that everything about water must already be known, right? Because water is so abundant all over. It's such a simple molecule. It's all over the place. And scientists have been studying water for, well, centuries. But I want to show you, just to start with, that although we think we know everything there is to know about water, I'm going to show 4 slides. In Seattle, we have a lot of clouds and a lot of rain, but this is not in Seattle. This is actually looking out over a vast body of water. And this is something that is not too infrequent. You see a cloud that's sitting here. But when you think about it, the water is evaporating from everywhere. It's evaporating from here, but also from here. And the question is, how is it possible that you get only one cloud? Admittedly, this doesn't happen all the time. When the water is evaporating from everywhere, is there some kind of force that pulls this water closer to the cloud, and therefore there's no cloud here, or what? Do you know the answer? Thank you for agreeing with me. No, it's not so clear, is it? Okay, so that's the first one. To demonstrate that although you think you know everything there is to know about water, you don't. Otherwise, you'd be able to explain this. So here's another one. As I said, in Seattle, it rains a lot. We have a lot of water. We all know that if you take a water droplet and drop it on some water, the two coalesce instantly, right? But this is a laboratory demonstration, and you can see that the droplets don't always coalesce instantly. They remain there, and you can actually see it on puddles sometimes. If it just rains, you see little droplets sitting on the top of the puddle. Any explanation? Don't all answer at once. Okay, I see a lot of nays. And here's another, this is a so-called water bridge, experiments done mainly by Elmar Fuchs in Holland. Very simple experiment, but the result is rather astounding. And so you take 2 beakers and you put them lip to lip and fill each one with water. And then you take 2 electrodes and put a high voltage between here and here, sticking the electrodes in. And when you do that, turn on the voltage, what happens is you get a bridge between the 2. It's interesting enough, but what's even more interesting is that if you mechanically move one beaker away from the other beaker, the bridge persists. And it persists indefinitely, and it persists for distances up to 3 or 4 centimeters. Explanations?
Magic.
No, I don't hear any explanation.
Magic.
Magic. Yeah, magic.
Surface tension.
Well, yeah, okay, all of the above. We'll talk about it a little bit. Okay, and finally, a 4th one. This one I like the best. This is a superconducting magnet. And the superconducting, this is a courtesy of Shogo Ueno, University of Tokyo. And right in front of the magnet, he places a trough of water. He puts in a little red dye for effect. We'll explain in a moment. And when he turns on the superconducting magnet, the water moves this way and this way, creating a space in the Red Sea through which Moses could walk. Explanation? Okay, it's astounding, I think. This is really amazing. So I started, for many years, we've been studying muscle contraction at the molecular level. And we did a lot of experiments and wrote a lot of papers. But I then came into contact about 20 years ago with a guy named Gilbert Ling, who, if you do the arithmetic, he's now 100 years old, alive and kicking. By the time I met him, he had already written 4 books or 5 books. And the main theme of these books, although there were several themes which made him unpopular, the main theme was that inside the cell, in your cells, my cells, inside the cell, the water molecules were not like the water molecules here, bouncing around on a femtosecond time scale and randomly oriented, but aligned with one another. He said that the water is a dipole, and all these dipoles are lined up inside of the cell, and that is really important. I got hold of several of his books, and it's tough reading. You need to know something about physical chemistry, and you need to be able to intuit what Gilbert really means when he writes, because his writing is perhaps designed properly for himself to understand, but others find difficulty. And so I was moved enough after giving his books to my students, and they came back to me and said, this is really incredibly important. And I thought so too, and so I was about to make a shift from the muscle contraction field to water, because I thought this is so basic, so fundamental, and if true, it's going to change everything. And my first task was to write a book, to try to translate Gilbert Ling into plain English, so to speak. And I did. I wrote the book, Cells, Gels, and the Engine of Life, and it presented the evidence not only that the water inside the cell was lined up, but also that I went somewhat beyond Gilbert Ling to describe that a transition in water from this ordered, aligned state to ordinary water was a trigger for many of the most fundamental cellular events. And I presented the evidence for that hypothesis. Well, the book got mixed reviews. A few people commented that it was well written and well presented. One guy from Harvard University said he wrote, quote, 303 or 4-page preface to the future of cell biology. And others said, this is the biggest nonsense I ever heard. This is trash. However, what I found after those mixed reviews is that discerning people will pick that book out, so, it's a natural. I guess maybe they like the cover or something. Well, I was so inspired after writing the book and learning about Gilbert Ling that I wanted to take the laboratory and transition from studying how muscles contract to studying water, especially water in cells. It's not so easy to study water. Epitomizes Gilbert Ling's idea. So here's a protein that sits inside the cell, and the surfaces of the proteins are mostly charged or hydrophilic. They expose the charges to the water that surrounds them. And Gilbert said, well, water is a dipole, and you can see that the dipoles will be stuck onto the charges on the surface. And if they have the right disposition, spacing and such, you get all the dipoles to stick onto them, and other water dipoles will stick to those, etc. And then you can get some order. And well, physical chemists agree that there may be some interfacial water that's ordered. Here, this has been studied for many years. The general consensus is that this ordering sticks out maybe 2 or 3 molecular layers. And after that, once you get far enough away, thermal motion tends to break up any kind of ordering. And Gilbert said, no, you guys are wrong. He said it could extend out many molecular layers, maybe dozens, or if not even hundreds of molecular layers. So Gilbert's ideas were disparate from the ideas of the mainstream, which put him, as you people well know, in great jeopardy in terms of his career, and he has suffered as a consequence. Anyway, if you have a region where the molecules are ordered, it's a crystalline-like or a crystal-like region, and crystals tend to exclude molecules and particles. For example, when water turns into ice, to get a pure crystal, any of the debris, particles, molecules is kicked out, and that's the only way you get a pure crystal. So this was like a crystal, and the basic concept that we were thinking is a fundamental aspect of that is if you have a region where the water is ordered, as Gilbert suggests, it excludes particles and solutes. So we're looking around for some kind of experimental preparation where we could see exclusion of solutes and particles. And we found it. This is a gel, ordinary gel, just like, for example, Jell-O, but we use different kinds of gels, placed in a chamber that you see here. And we added water plus particles. And the particles that we used, routinely we used, they're called microspheres. They're actually little spheres. You can get them functionalized in different ways, typically 1 micrometer in diameter. You can get smaller or bigger. And we put the particles in the chamber with the water, suspended in the water, and lo and behold, we found a region. This region is about 10 micrometers, which by molecular standards is, pretty substantial. It looked like this is a region that excluded the microspheres, and we thought, maybe it's possible that next to the gel, next to the surface of the gel, the water molecules are lining up. And the situation was actually more dramatic than that because we did videos. One of the first videos we took, we could see that something was growing from the boundary and pushing those microspheres away until they finally stopped. And here we're looking at 50 micrometers, which is maybe half the thickness of one of your hairs, not mine because I don't have too many left, but so pretty substantial. We became obviously interested in thinking that maybe there's some kind of interfacial effect that grows and pushes out those particles. So we got excited about that, and after we did many experiments, and a friend from Australia said, you got to give this a name. We thought this was an original observation, and he said, why don't you just call it the exclusion zone, because it excludes, and that made a lot of sense, and the abbreviation EZ really works well because it's easy to remember. It doesn't work in Europe because it's EZ. That's a problem, but it works. And so we call it EZ. It's one of the several names for this zone. Another example, because we began seeing it next to a couple of different gels, we wanted to look at a polymer. And a polymer that we came upon because of some quirk is a polymer called naphion. And naphion is a bit like Teflon. It has a Teflon-like backbone, but sulfonate negatively charged groups that stick out, many of them. It comes in a sheet. And so we took the sheet and we cut out a little piece of the sheet, arrowhead form, and we plunked the sheet at the bottom of the chamber. And we poured in water plus microspheres. And as you can see, already some exclusion zones opening up, and if we watch it over time, this is actually about 10 minutes compressed, it builds, and it builds in this case to something like 0.5 millimeter. It's pretty big, that's like 500 micrometers. We thought, well, the finding, a lot of people got interested in this, and this list was created, I think 5 or 6 years ago. These are some of the laboratories that have confirmed it. By now, many more. It turns out it's not original. It turns out in 1970 in the Journal of Physiology, London, some investigators were studying the lens of the eye, natural lens and artificial lenses, and they used microspheres and other particles, and they found it's exactly the same finding, even the same size roughly as we found. So it's not a new observation, it's an old one. And so what I want to do is get to the basis of all this, and in order to do so, I want to answer 5 questions. Okay, the first question is the exclusion phenomenon general, or just those couple of cases that I showed? Does it really arise from the ordering of water? Because all I showed was a zone that's particle-free. There's no evidence that it had anything to do with ordered water. And of course the answer is going to be yes. And the third one is, can water ordering explain those first 4 slides, or observations similar to those first 4 slides which you couldn't explain? What energy creates this order? As you all know, to create order from chaos, you need to put in energy. And where does the energy come from? The chamber is not plugged into the receptacle or anything like that. question that stumped us for a while. And might these findings apply broadly? I have an asterisk on the second one, right here. And I want to, when I talk about that, I'd like to include a discussion of water memory. Some of you may have heard about some debacles that took place about 30 years ago about the memory of water. And I want to address that because I think there actually is some basis in what we've uncovered that could explain the idea that water can actually contain information. Okay, question one, generality. So, two questions that you might ask. What kinds of surfaces create the exclusion zone? And what's excluded from the exclusion zone? So in terms of surfaces, we tried numerous gels. I think we must be over 20 or so. Every one, hydrogels, gels made of water and some other material, and they all showed it. Polymers, we found that many, but not all polymers, they had to be hydrophilic in order to see this. And even some hydrophilic ones didn't show exclusion zones, but many of them did. So it was fairly general. We tried biological surfaces, and we've tried half dozen so far, and they all show EZs. And we even tried monolayers, that is a single molecular layer on gold. It's a standard procedure, and it's one molecular layer. And we found just by putting one molecular layer, we could get a giant exclusion zone, 200 micrometers, which means, the way we interpret it, is that this single monolayer acts as a template, and the template can allow the buildup of the EZ. So it's pretty general. And now what's excluded? We tried so many different things, starting from big particles to small particles to big molecules to small molecules, and essentially, I won't say everything is excluded, but almost everything we found is excluded. We found even that water was excluded. So you take something with an EZ and you pour water, and the water doesn't get through; it stays on top. So we're down to very small molecules, including some evidence that salt molecules are excluded as well. That's not definitive, but we think so. So in terms of generality, many hydrophilic surfaces generate exclusion zones, and many solutes are excluded. So it's pretty general. Question number two. Is this zone physically different from bulk water? I've suggested it might be, but I provide no evidence. What's the evidence? So what I want to do is I'm going to list 8 pieces of evidence. By now there are, I would say, a dozen or more. And I'm not going to go through details of each one because most of it's been published and anybody who's interested. So I just want to give you an idea. And I will focus on one of these pieces of evidence because it's very important. The first one is that the water molecules in the EZ are more constrained than water molecules in a glass. That's using NMR, nuclear magnetic resonance. And the EZ molecules are more stable. If you measure the amount of infrared radiation coming from this zone, it's much reduced compared to ordinary water. It looks like it's more stable. It's not generating, the charge is not moving a whole lot. It's not generating much infrared radiation. We found that the EZ typically has negative charge, is not neutral. And this was a big finding for us, and I'll describe it in a few moments, because you expect anything dealing with water should be neutral, but this is not neutral. We have, I think, good evidence for that. We found that it absorbed light at a wavelength of 270 nanometers. That's in the UV range. Ordinary water doesn't do that. We found that it's more viscous than ordinary water. The molecules are aligned in it. The molecular structure differs from ordinary water. And finally, at least on this list, the optical properties differ from ordinary water. That is, the refractive index is higher. And it's funny because This was studied not by us, but by two Russian groups, both in Moscow, never met one another. One passed after he wrote the paper, so they maybe will meet each other on the other side. But they both got the same result, one in the biological system and one in the physical system. What they found was that the exclusion zone was up to 11% higher in refractive index than ordinary water. Okay, so I don't want to belabor this, but I do want to talk about the negative charge. So how do we know that the EZ has a negative charge? Well, so we set up in a chamber, we took a piece of Naphion, which builds nice EZs. We use Naphion quite a lot, although not exclusively. And we wanted to see the EZ first of all, and to see it, what we do is we put some dye in the water. So this is water here. This is a chamber, we're looking from up above, and we have water that's here. And we add to the water pH-sensitive dye. This is for another. We could have used any dye that we wanted to that gets excluded, and it seems they almost all do. So we can see where the exclusion zone was. I'll come back to the different color distribution in a moment. But we took microelectrodes. These are electrodes that taper down to about 1 micrometer. Incidentally, parenthetically, developed by the same Gilbert Lynn. He should have gotten the Nobel Prize for that, because this has become a really important piece of apparatus used to measure the electrical potential insides of cells. It was a breakthrough and still is. Okay, so we put one electrode here and one electrode out here somewhere, and we confirmed that there's negative charge. The electrical potential was typically 100, 150 millivolts negative, which incidentally, those of you who are biologists, maybe some, this is the electrical potential close to it of the inside of the cell. Not, I think, by coincidence. So this is negatively charged. When we first got the result, we were a little skeptical. We did it for fun. We really didn't expect negative charge here. And my friend Felix from Russia was with us, and we were both a little skeptical of this result. So he made a phone call to his wife. He said, "Honey," or whatever is the Russian equivalent, I don't know, "could you make this measurement? Here's what we got." And it just happened that his wife was dealing with gels and microelectrodes, so she could easily do it. And within a few days, she said, "Yeah, I got the same result." This was with similar result, but still, you know, we were skeptical because it doesn't make sense. You know, the experiment, we got a chamber here, and we put a piece of Naphion or gel or something that nucleates the EZ, and you take water, and the water is negative, the water is neutral, and you take the water and you pour it into the chamber. How is it possible that you get a large region of negative charge? So we're scratching our heads because it didn't make sense. After a few weeks, we determined that the only way this could happen that we could think of is that the water molecule is getting split. You know, H2O or H plus, OH minus gets split into the H plus and the OH minus, and somehow the negative ones are accumulating here. And if so, there must be positive ones somewhere here, if that hypothesis is tenable. So, but what's the evidence that there are positive charges out here? Well, the evidence is right here in front of you, because this is a pH-sensitive dye, and the color orange corresponds to a pH of 3 or less. So this is just full of positively charged protons here. So indeed, it looked as though this was a tenable hypothesis, because we got negative charge here, and we got positive charge here. So it made sense that the water molecule was getting split. And this is not a radical idea, because in photosynthesis, I think it's a 20-step process. The first step is the splitting of water into H plus and OH minus, so there's plenty of precedent for that. Of course, we weren't sure, and so we did an experiment. We stuck one electrode in the negative part, another one in the positive part, connected the two electrodes with a resistor, and we expected, if really there's positive charge here and negative charge here, you'd expect current flow through the resistor from here to here. And we did the test, and this is one of the first records that we got. We have many. this is current and this axis versus time. And you can see it starts at a kind of so-called high level and goes down, but it doesn't go to zero. Zero is here, it goes down to some plateau level. So there's a steady current flow from plus to minus, which told us that our idea that the water molecule is getting split into positive zone, negative zone made sense. There's a charge separation that occurs. And we were pleased with that result. You know, just a comment, it has to do with our own pet ideas and hypotheses. This was a guy who was famous in the muscle contraction field, and I still remember one comment he made. He said, regarding our theories and ideas that we all love so much, he said, only artists love their models more than scientists. So we need to pay attention to that. Anyway, so what we've discovered there is essentially a charged battery in water. We have a surface of some sort, some hydrophilic surface next to water, and this region becomes the EZ with negative charge, and there's ordinary water with lots of protons in it. It's a battery, basically. And I'll show you some applications of that battery a little bit later. So far, the summary is we have some kind of liquid crystalline water zone that exists next to hydrophilic surfaces. It's got negative charge. It's not neutral, okay? It's got negative charge. It excludes solutes profoundly. And the next point, which may or may not have been obvious, but it will be soon, is that the idea of dipoles, I think, is wrong. We were very happy to have these nice pictures, which appeared in my 2001 book, but there's evidence that it's simply wrong. I'll tell you why in a moment. And the region of order may extend very far from this kind of surface. And how far is very far? If you do the arithmetic on how big it is, and a typical water layer will be about 1/3 of a nanometer, you can compute that we're talking about millions, up to millions of molecular layers, not 2 or 3 or 10 or 30, but millions. It's a lot. If the evidence is correct, then that's what we have. Long ago, it was suggested that water might have a fourth phase. And the premier exponent of that was Sir William Hardy, more than 100 years ago, who was a famous physical and colloid chemist. He said, we got a problem. He said, there are so many anomalies of water, even 100 years ago. Now, if anybody examines Martin Chaplin's well-known website, where he shows something like 60 different anomalies of water, things that don't fit the theory. That's an anomaly, right? But 100 years ago, he said, even then, there were many known anomalies. And he said, look, you know, there are too many anomalies, something's not working. The idea of 3 phases of solid, liquid, vapor just doesn't answer all the questions. And he was suggesting that there might be a 4th phase of water somewhere in between a solid and a liquid. So it's not a new idea, and so we tend to, I don't know, justified or not, but we tend to call this exclusion zone, the millions of layers, up to millions, another phase of water, a 4th phase of water, a phase where the molecules are somehow ordered. Now, what's wrong with that dipole structure? It's so nice, it's so pretty, so simple, so easy. Well, the problem is actually elementary. So, this region has negative charge. It was confirmed by us, by the Russian woman, and by us again, using a different surface. So, and the problem is that we found negative charge, but dipoles are neutral. So you can line up dipoles from here to the moon, and you'll never get negative charge. You get neutrality. But the experimental results showed negative charge, so it can't be right. Sorry, nice picture, but so we went through several years of thinking and developing, and I don't want to go through the process because it takes too long, and there's more stuff that I want to tell you about. But here's what we came up with, and it's described in my recent book, not the one I showed, but the fourth phase book. I'll show a picture in a moment. All the reasoning that went into it. And the easy structure we come up with is this. So you have a surface, a material that is one that's capable of generating EZ, some hydrophilic surface, next to water. And what happens is the EZ layers grow in sheets. These are hexagonal or honeycomb sheets. And they grow one at a time. So you see something like this and this. And they just keep growing one sheet at a time until it stops growing. And if you look at the structure of one of these sheets, it looks like this. It's a honeycomb. It's a hexagonal, and as you know, nature likes hexagons. And there's much evidence for hexagonal structures of water, especially interfacial water, even ice. And it consists of the red oxygens and the blue hydrogens lined up this way. And if you were to take a unit cell and count the number of hydrogens and oxygens, it doesn't turn out to be H2O. And nor would you expect it to be H2O, because H2O is neutral. This is not neutral, it's negatively charged. And what we found, if we do the counting, is actually H3O2. And it makes sense that, this actually makes some sense, because if it were H4O2, it would be double this one, and it's neutral. So by missing one positive charge here, the net charge is negative. See, so that makes sense. And so these pile up. Okay, so we have some kind of liquid crystalline structure of the EZ, or fourth phase. And a question that arises, this is a slight digression, but I think it's an important one, this is a liquid crystalline structure. And liquid crystalline structures can often be solidified. Example of a sugar solution, and I think it was Louis Pasteur who found that this bends light, and so it's polarized in some way. And it appears that the sugar solution creates a kind of liquid crystal. And you know, if you put a string in here and you wait long enough, then you get some rock candy, which is basically solid sugar, crystals of sugar. So you've taken something that's a liquid crystal and it's solidified. And the question arises, we've got liquid crystalline EZ, can it be solidified? Now, you've never heard of some kind of water being solid at room temperature, but we have it. I'll show you. And the first person to do it is Vittorio Elia from Naples. Well, it wasn't exactly Vittorio. His wife, Elena, does the experiments, and it took many experiments to repeats of the same thing to get something major. So what they did was, they took a sheet, they took a sheet of Naphthalene, put some water in, and the EZ grows right next to the face of the sheet. It starts growing up. And she took a roller and rolled the EZ off, and of course you can't just get EZ, you'll get some ordinary water, and collected that, and then poured more water, did it again 10 minutes later, etc., etc. She kept doing it many times, many cubed, or to the fourth power, something like that. And if she dumped it, or they dumped it into a beaker, after some time, you got a structure like this. And so this is clear water, as you might expect, but right here is a cloud-like structure that presumably contains the EZ material. You wouldn't get this if you didn't have, if you had pure water. So this presumably EZ, and it's not a coincidence that it looks like the clouds in the sky, because I think the clouds in the sky, different subject, also contain EZ water. But the real experiment they did to get the solid is not this one, but they took the water and they put it into a flask, and they put it through a lyophilizer, which is a device that protein chemists use to get rid of water, free water, and then took it out and brought it back to room temperature. This is what they got. This is a residue that's sitting at the bottom, and they could scrape it out. They could do mass spectrometry to find out that there are no serious contaminants. This is basically a kind of water. that's here. They gave us some too, put it into a little flask, and I was a little embarrassed to tell them that I gave little bits of it to each of the people in the lab, hoping they would do something, but nobody ever did anything. So we began repeating their experiments, because I think, for me, it's like totally amazing to have a kind of water that's a solid at room temperature. You know, we know, we put it in the freezer, we have a solid. This is room temperature. So we repeated their experiments, and we're just doing it right now, and we've had some success. And this is, it's just a little bit of it, because it takes a lot of experiments to get enough material. And we found that it shows the 270 nanometer absorption that we expect. So why am I showing you all this? Well, number one, I think it's amazing. Number two, there's a possibility of thinking about this solid substance, or even the liquid crystalline EZ, as containing memory. This is water memory. Now, I don't know if any of you have come across this idea of water memory. No? Anybody? Yeah, a few. Okay, so you know the story of Jacques Benveniste and his problems with water memory. It's inconceivable that this can store information. And by now, when Jacques Benveniste did experiments demonstrating that, he lost his career. A committee was sent by Nature to investigate what he was doing, and they said it was a trick. They never could identify the trick, but by now, this has been repeated many times. And this guy has become a hero at our annual water conference, because, well, he was essentially crucified. Many people are now reporting that water can store information and give out information. So among the people who I associate with at the conference, this is like standard material by now. Even Luc Montagnier won the Nobel Prize for discovering HIV, has switched now to studying water memory and information in water, and he's able to take some DNA sitting next to water. He says the information from the DNA is transmitted, maybe electromagnetically or something, to the water, and then he uses that water, just the water, to create new DNA, and the new DNA has the same sequence of the DNA that was sitting next to the flask of water. Amazing. So there's a lot of stuff going on, and I raise this for a number of reasons. One of them is when we think of memory, so we have to think of a memory stick, and it's pretty dense with information carrying atoms, and typically looks something like this, with silicon atoms arrayed in a 2- or 3-dimensional lattice. Each one is regularly positioned. And as you know, if you can address one of these, it's either on or off, or 0 or 1. It's 2 different states, and that gives you information. Now, if you look at the EZ lattice, it's very similar, because we have oxygens and hydrogens that are situated regularly, both in this direction and also in the Z direction. So, and what's really interesting about it is when you think about how information could be stored, you have electromagnetic waves that are coming in, and suppose an electromagnetic wave impacts certain oxygen atoms, but maybe not others, depending on their position, something like this. It turns out that each oxygen has not 2 states, but actually 5 states, 5 oxidation states. The one we know about usually is -2. There's -1, 0, +1, +2. So if you think about, if you calculate, if you could address, and we haven't figured out how to do that yet, if you could address each oxygen atom, then the memory storage capacity of this, we calculated, because it had 5 states instead of 2, is something like, I forget the number, 10 to the 8th or 10 to the 9th, more than a standard computer memory. So the potential memory density is qualitatively higher. So, you know, with 5 oxidation states, huge information storage capacity, the question arises, will information storage in EZ water eventually replace conventional information storage? We don't know that, but we think it's a possibility, at least theoretically. Now, some of you know this guy, Masaru Emoto. 1, 2, 3, 4. Okay, so let me explain. So Emoto, who passed in 2014, but his group is being perpetuated by his son Hiro and several other highly dedicated people. And what he demonstrated, he was a healer to start with, and he found out that water was really important, that he could convey information to the water, either by focusing his intention on the water or by playing music to the water. And when he froze the water to produce crystals, the crystals were either beautiful in some cases or ugly in other cases. And the reason that almost no scientists pay any attention to it is that he cherry-picked his data. So he would pray to the water and get multiple crystals, and he'd pull out the one that looked the best to demonstrate his point. That's not something we would do, but that's what he did, and he claimed he's a spiritualist and he's not a scientist. Right now, some of the people in the organization are doing this in a more scientific way. But here's an example of the kind of thing that he found. So if he played a Mozart symphony and froze the water, it looked like this. John Lennon, and like this. If he thought or expressed the word, I'm not sure which, about peace and thank you, beautiful ice crystals. But I think this means you fool, or something like that. Is that, no, what does it mean?
It's like nauseous.
Nauseous?
Like, yeah, when you reveal something, like something makes you nauseous.
Sickening.
Sickening. Oh,
Yeah.
sickening.
I think
Sickening.
that would be good.
Okay, and you can see the ugliness and heavy metal music, if you will, would look like this. And so scientists are obviously highly skeptical. And here's a picture of him after Fukushima, and there are many people actually around the world who are continuing his work and showing ugly and beautiful crystals. Akiko is working in Germany doing it, so it's quite around the world, and there's a big peace movement going on. And what I want to say is just that the experiments that we've done and the model that we've come up with has capability of storing information. We're not standing up and saying that everything that Emoto did is correct, but the basis for this is there. We also found, by the way, I don't want to take the time to, but if you want to freeze water, we found that you must go through it, sorry, let me back up a moment. The structure of EZ is not so different from the structure of ice. It's different, but it has the same kind of hexagonal motif. And we did experiments published that show that if you freeze water, if you take this water and want to produce ice, you first must go through the EZ state. And the EZ state then changes to ice. And we have evidence published for this. I'm sorry, I'm digressing a bit, but the point is that this goes through some stage where there's a capacity for information storage before it becomes ice. Okay, so that was a digression, but the answer anyway to question number 2 is the EZ physically distinct from the bulk. And I think the answer is yes. It's physically distinct. The best evidence we have is that it's a layered honeycomb structure, and it has information storage capability. Okay, question 3, can crystalline water explain counterintuitive anomalies? The kinds of slides I started with. And I want to just present a couple for you, not necessarily all of the ones that I showed initially because I don't have the time, but here's one. Okay, so I think many of you have had the experience of taking a glass of water and taking a pin or a paper clip, putting it on the top, and it floats. Shouldn't float because the density is higher than that of water. And if you put it beneath the surface, it plummets right to the bottom. But carefully placed, it'll stay there. And here's another example of such. This is an old Hungarian coin that does the same thing. So the typical response is, oh yeah, well, water is known to have a high surface tension, and that's why this floats. But the high surface tension is thought to occur at one molecular layer. So water molecules like to hang on to neighboring water molecules with the so-called hydrogen bonds. They make and break and such. And they'll do that, but the guys sitting at the top have a problem because they want to reach up and, oh, there's air up there, no water molecules, and so a potential hydrogen bond will flip down, and therefore the top layer of molecules will have more connections to neighboring molecules and therefore should be stiffer. That's one molecular layer. So the question is one molecular layer sufficient to do this? We thought maybe not, and we did experiments to show that the liquid crystalline or EZ water grows at the air-water interface. It's not just next to hydrophilic materials. And the experiment to demonstrate it is shown here. So we take a chamber, and the chamber is made of two pieces of glass, and the glass, they're put near one another to make a chamber and sealed around the edges. So we have a chamber. We pour water and microspheres into the chamber, and what you get is, well, you have air up here, you have a meniscus that forms here, and this is cloudy because the microspheres scatter light. And we found that over about 15 minutes or so, that a clear zone opens up and then remains stable. So this clear zone is similar to an EZ because it's free of microspheres. We took an electrode and measured the electrical potential, and we found it's negative, just like most EZs. And the next slide will show you that this clear zone here is not water. It doesn't behave like water. It behaves more like a thick gel, like a thick rubber band running along from here. And you can see that here. So this is the same as before. Here are the microspheres in water. And here is the clear zone near the top. And we're going to take a glass rod, touch the top, perturb it mechanically, move it back and forth. And what you'll see is that this is a cohesive region that sticks together, and despite these mechanical perturbations, the width of this doesn't change at all, or at least not noticeably. So here's coming down, lift up, and go back and forth, and you see the height of that dark zone remains constant, as though it's some rubber band-like material that runs back and forth, or like a gel, in the way that EZ should be. And so we conclude from that what keeps this up is not a single molecular layer, but many structured layers that create the high surface tension, if you will. That's what keeps it up. Essentially, you've got EZ at the top. And that may explain this. Some of you have seen this before in National Geographic. This is a lizard. Lizard lives in Central America. And mostly it spends its time sitting on a tree branch. But when it gets excited or whatever, jumps into the water and walks on the water. You see, and it's because it walks on water, it's called the Jesus Christ lizard. And you can see what it does. And so we think it's not just a single molecular layer that can do this, but a thick layer. So that's one explanation for what this creature does. The second example is crystals. As you know, some crystals can be pretty stiff. The liquid crystals are generally not so stiff, but they can be stiff. And then those of you who have gems of some sort, diamonds, rubies, whatever, they're crystals, and they're really hard. So if you think now about this water bridge, by the way, this is called the floating water bridge because when Elmar Fuchs came to Seattle and saw the floating bridges that span Lake Washington, maybe you've seen them, he began calling the water bridge the floating water bridge. So now it's called the floating water bridge, I think after our water bridges. So ordinary water doesn't do that. I mean, you've never seen water doing this, but it does it. And the possibility is that the liquid crystalline EZ water that's here becomes stiff enough because of the voltage that's applied that it forms a stiff bridge. It's so stiff, you can measure the stiffness by the droop, the diameter, and the span. It looks like it's almost stiff enough that you could walk across it. So I haven't presented many anomalies to you because of time. But I think the answer to question 3 is yes, liquid crystalline water can explain various anomalies. So now the $64 question, what charges this battery? We couldn't figure it out. I mean, in order to create charge separation, just like your cell phone battery, and to create order, they both need energy. So where on earth does the energy come from? And I'm a little embarrassed to say that for 2 or 3 years, we couldn't figure it out. Question?
No, what about the static on the hydrophilic substance?
Once more?
The static attached to the hydrophilic substance.
You mean like a surface charge to create the energy?
Yeah, could that be?
Yeah, it could be, but could you explain 1,000,000 layers that way? I could imagine like surface energy creating one layer, but what about the next layer and the next layer and the next layer? I'll give you the answer that we came up with, but you know, we can discuss later. You may have a good point. Well, so I'll tell you the experiment that was done, but first of all, I asked this question. I was giving a class, and I was telling them about some of our findings, and I asked the class, where does the energy come from? And of course, there were a lot of blank stares, and one student tentatively raises his hand, and he says, light? And I quickly hired this student to work in the laboratory. He was great. He got what we found to be the correct answer. It's incident radiant energy. And so how did we find out about that? Well, so a student was doing an experiment, and he was doing something that he should not have done, but students like to do what they want instead of what I suggest they do. So he's got a bench sitting here, and he's got a chamber, and he's looking with a microscope at the exclusion zone. And next to him is a lamp, is a gooseneck lamp. And for fun, he took the gooseneck lamp, he shined it on the chamber, and this is really what he saw. This is original data, so, and the lamp is not, but, so he's shining the lamp. This is a piece of naphion sitting here. This is an exclusion zone, and all the microspheres are here. And usually the exclusion zone runs straight along here, a narrow band, or wide, depending on your scale. And when he was shining the light, he could see the exclusion zone growing. And then I suggested to him, well, remove the light. And when he removed the light, it collapsed back down. It took, with a time constant of some tens of minutes, but it went back to the original. So, you know, it didn't take a rocket scientist to figure out that you turn on the light and it grows, so that maybe photons are responsible for the growth, the energy coming from photons. We did a series of experiments. I won't bother, it's all published, to check which wavelengths were the most interesting ones or most effective. And we started with in the UV range, and used a series of discrete wavelengths running through the visible all the way through the infrared up to about 4 or 5 micrometers. And we found that by far the most effective was infrared, and particularly 3 micrometers. It was, if I were to say, perhaps hundreds of times more effective than the visible wavelengths. So the question always arises, well, infrared, that's kind of a weird kind of energy. Where do you find it? Where does it come from? And the answer is it's all over the place, that everything you have is radiating infrared energy. And if we could close the shades, turn off all the lights, and so dark that you could see nothing, take out your infrared camera, like a regular camera with an infrared sensor, you get a beautiful image of everything, everybody, the tables, your glasses, your briefcase, etc. It's all over. And sunlight too, approximately half of the wavelengths that come through, about half the energy is in the infrared region. So it's all over the place. It's not just looking in your oven and with the orange glowing elements that are on, where you feel the heat and you say, oh yeah, heat, infrared. It's all over. And because it's all over, it means that anytime that you have the right kind of hydrophilic material sitting next to water, you'll have a certain amount of EZ water that grows based on the amount of ambient infrared energy, and some visible too, but mostly infrared energy. And if you were to increase the amount by turning on a lamp, then what happens? It grows. And if you take away the extra, it goes back to the original. So the answer to question 4 turns out very simple. This may not be the only answer, but we know that EZ buildup is powered by light, photonic energy, particularly in the infrared region, which orders the water and charges the water battery. So it's like sort of going to Hawaii. You just lie and absorb the light, and you get energy. And so a question that arises from this is, well, gee, if you can just get energy by sitting in the light and in the sunlight, if this is building energy, at some point it should explode, or there ought to be some way to release the energy. And how is it getting released? One way is it could just re-radiate energy, but the natural question is, all that energy that's being absorbed, can it be harvested in some way? And I'm sure that there's nobody, maybe I'm not sure, because this is a unique group of people, but have never seen a glass of water doing work. Maybe I'm wrong, but most people have never seen a glass of water doing work. I'll show you an example of a glass of water doing work. So the answer is, I think yes, the energy that comes in can be harvested, just like plants, So the experiment was done by another disobedient undergraduate student. And we had just found out, there have been quite a few working in the lab, and it seems they're the most open-minded, and they're the ones who find interesting stuff. So this student, we had just found out that naphion comes not only in sheets that we had been studying, but also in tubes, looked like this, like a straw. And I asked him to check to see whether if you take this tube and put it in water with microspheres, can you find an exclusion zone right on the outside and also on the inside? We expect annular exclusion zones. And he was quickly able to find that, but one day he came, I was sitting in my office, and I was sitting with a visitor. I can't remember who the visitor was, maybe it wasn't too important. And the student comes barging in. Didn't knock or anything, just comes barging in. He said, I found something that looks interesting and I thought you'd like to know. So what did he find that's so interesting? He said, I've been looking with the microscope, and what I see is that the water keeps going through the tube endlessly. It just keeps going and going and going. And I thought, if this is right, if this is true, it's amazing, because you need energy to drive something through a pipe, like your heart driving fluid through your large arteries and such. And there's no pressure gradient like developed by the heart here, because these are sitting at the same vertical level, and yet there's flow going on. So, you need energy to do it. And the only energy we could think of is that we knew by this point that the water is absorbing energy from the environment, especially infrared energy. And that infrared energy's got to do something. And this is what it does. And we quickly checked to find that, indeed, the experiment was okay. He didn't flub in some way. So the experiment looks something like this. You take a little piece of tube, you fill it first with water and microspheres, and then you put it into this trough or chamber with water and microspheres again, and you take it to the microscope. Green light, we minimize the amount of light, and look in and see what happens. And what happens looks just like this. So there's flow that goes through, and we've had it going for as much as 1.5 days without stopping. Someone said, you used a naphion tube. Maybe there's something about naphion tubes, something special or something weird about naphion tubes. Why don't you try other kinds of tubes? Where do you find small tubes made of hydrophilic materials? You probably can, but we couldn't, so we made our own. How do we make it? This was another ingenious idea of one of the students. take a gel, and as the gel is gelling, you put a wire in, and when it gels, as it's gelling, pull out the wire, and you're left with a tunnel. So we had a gel that was kind of a block like this with a tunnel running through it. And we take this block of gel with a tunnel, and we put it in a chamber that has water and microspheres. So to see if we got the same effect, because these are different materials, they're not Nafion. So here's a polyacrylic acid gel, here's the gel, here's the tunnel running from here to here. And you can see, as expected, that next to the gel here you find an exclusion zone, and next to the gel you find an exclusion zone. And all the microspheres are pushed beyond those two exclusion zones to be right in the middle. And if you look at the video, same effect. We've done this now with a half dozen different gels, and you get the same result, except the speed is different with different gels. So another person came to us, we get a lot of input, feedback, and said, well, you think all of this is built by light, that light energy. You could do a simple experiment, just turn up the light and see if it goes faster, right? Brilliantly simple. So we did the experiment, and we published the result, and the result shows that we could get 5 times speed increase just by turning up the light. So this is definitely powered by light. So essentially, we have a hollow tube sitting in water, and water flows through it. And if water is flowing, work is done because the water has viscosity. And to do work, unless this concept is wrong, you need an energy of some sort. And the only energy that we can envision, unless someone has another idea, is what we already found, that the energy absorption from the environment, that's a necessary condition for this, because we don't know of any other kind of energy input. And so basically, if that's correct, it means that water transduces light energy into mechanical energy. So this water is a transducer of light energy into mechanical energy. The water is doing work. It sounds weird, but think about what goes on in the plant in your office, on the windowsill, right? Where does its energy come from? Well, it comes from light, see? And light, step 1 in photosynthesis, as I mentioned, is the splitting of water into plus and minus components, very similar to what I presented. And it's possible that what I presented would actually apply even to the plants, because there are some unknown and inconsistent features of understanding of photosynthesis. Anyway, builds chemical potential. The chemical potential then is responsible for metabolism, for growth, for bending, for essentially all that the plant does comes from energy from light. And I'm suggesting to you from our experiments that the same thing happens in water. And it's not a surprise, because the plant is mostly water, you see. So we're suggesting that the same thing occurs. So we come up with this equation, and I know that you know an iconic equation that looks a little bit like this, but this is different. And essentially, what we're suggesting is that whenever you have water, you have energy. Water is not in equilibrium with the environment. It actually has energy of its own, and the energy ultimately comes from light. I know the units don't match, and please forgive me for that, but all that we need to do is put a K somewhere. Okay, and finally, question 5. Why is all of this important? I think it could be if what we found is correct. Quite a few other groups have gotten involved and have confirmed various of what we found. It could be foundational for any or all science involving water and molecules and light. And there are many things that could be discussed, and we could be here all afternoon and tomorrow. But also, what I do want to focus on is a small segment of this, and that all of what I presented to you is foundational for your health. Now, I'm sure that's not obvious at the moment, but I want to tell you why I think that this easy water is absolutely, it fills your body and it's foundational for your health. So, to get into that, the first question is, does human body use radiant energy? And most of you will think no, or some of you might think yes. So, I mean, we receive radiant energy, and so there are two possibilities. One is that we re-radiate it out, and the second is that we actually use it. Suppose you were Mother Nature, and you did pretty well with green plants using photosynthesis, and also some unicellular organisms that also use light for their energy. And one day you decide, you know, you've had a lot of experience, you want to do something new, why don't we invent animals? Okay, so if you do that, and you're as wise as we suppose is Mother Nature, are you going to throw away the mechanism that uses light because animals can forage, get their energy through food, or you're going to keep it as a supplement or in reserve or something like that? I don't know about you, but if I were Mother Nature or Father Nature or unisex nature, whatever, I'd want to keep it in reserve, right? Because this energy is coming in, we're absorbing it. Why would we abandon this mechanism? And so, first idea that came to mind is the cardiovascular system. Is it possible, because the capillaries are pretty superficial, light can come in, and depending on the wavelength, it can penetrate reasonably far. Is it possible that the light is somehow powering the cardiovascular system in some way? Now, at first it sounds preposterous, and it sounded that way to me as well, because I started my career studying the cardiovascular system, and it was clear to me and everybody else who studied it from the time of William Harvey that the heart, the contraction of the heart builds pressure and drives the blood through the arteries. And I thought we had it all wrapped up. I did my PhD thesis simulating the cardiac contraction and the pressure and the flow throughout the cardiovascular system. And I was pretty sure we had it nailed, that there were no lingering problems. Until I took a trip to Moscow, and my friend Vladimir Vyakov at Moscow University introduced me to the guy in the laboratory next door. No English, but good translation. And the guy was telling me that there's a big problem in the cardiovascular system. And in somewhat maybe a condescending way, because I thought I knew all there was to know about the pressure and flow, I said, tell me, what's the big problem? And he said, oh, it's very simple. I said, oh, really? Oh, simple. He said, the problem is this. He said, the problem, it's a plumbing problem. So the red blood cell that passes through the capillaries is 6 or 7 micrometers in diameter. And in healthy young adults, the capillaries go down to 3 micrometers, 3 or 4 micrometers in diameter. How does a lumping red blood cell work its way through? So he said, you know, it requires energy. Something has to push it through. And what happens, I'll show you in the next slide, is that the red blood cells bend as they go through. You have to crunch them down in order to push them through. And it looks like Mother Nature screwed up on this design. It's sort of like, you know, like a toilet. Sometimes you have to take the plunger and push everything through to make it work. It's the same problem. He calculated, he said, in order for this, if the heart were responsible for sending the blood through the capillaries, he calculated, and everybody knows that calculations can be wrong, he said the heart would need to develop a pressure that's something like 1 million times the pressure it develops. Well, that's really high blood pressure. So he said, there's got to be another energy source. And I was actually surprised to find various people, books and such, and people saying the heart is not really a pump. I never could quite understand their point, but maybe I need to study further. But it's clear that the heart is generating pressure, but the pressure seems to be not enough to push the red blood cells through the capillaries. So the Russian guy was telling me about various ideas, and I found myself unable to listen too much to his ideas because I thought immediately, I think I know what could be the answer, and it has to do with the few slides that I showed before about getting spontaneous flow through light energy. So, but if you look at typical, this is, I forget which species it is. I think it's the bat wing, and here you see muscles, and these are capillaries that run through, and you can see that the red blood cells are compressed. They're squeezed in order to go through, and they go through. And this guy is having a hard time. They can get it really, really squeezed and compressed. And so, and also you notice that there's no beating. This seems completely decoupled from the heart. So it seems that some other mechanism needs to be. We know the red blood cells are supposed to look like this, but they don't look like that. So I thought immediately, we have an experiment, and the one that I just showed, hollow tube, radiant energy drives flow. And the question is, might radiant energy help to drive blood flow? And I was immediately enamored of this idea. And my student, who's been working on the project, just finished his successfully defending his thesis, and the next slide after this one shows his main result. But we looked into the literature, and we found that there are actually something like a half dozen reports over the past 50 or 60 years that demonstrate that after the heart stops beating, flow continues. Now that's weird, because we all expect we're dead. But it's interesting that measurements using, each of these experiments is done differently, and all come to the same conclusion. And sometimes hours after the heart stops beating, there's still flow. And the one study that really impressed us was one published about 5 or 6 years ago, an Israeli study on mice. And they were measuring capillary blood flow using a powerful optical technique to see the capillaries, optical coherence tomography. And so they reported that they're doing the experiment on the mouse, and after they finished their experiment, they sacrificed the mouse by clamping the aorta. And the mouse is dead within 10 seconds. The heart stops beating. But the flow continues. They've got their flow meter recording flow. Of course, it's a reduced velocity, but it just keeps going. And they measured 10 minutes, 20 minutes, 30 minutes, more than 1 hour before it stopped. They repeated the experiments using 10 different mice, and they got the same result each time. So, blood flow continues after the heart stops beating. There must be some other driving mechanism that's responsible for it, a mechanism that maybe operates also when the heart is beating as well. So Lee, my student, did the experiment. He used a chick embryo, fertilized at 3 days after fertilization. So here's the egg, and you could lop off the eggshell, and then with the microscope you can see, and here's the yolk, and you can see the embryo that starts to grow. These are all blood vessels coming out. And he could easily image a region here and a region here. And his experiment was to say, if I stop the heart, and he could stop the heart by injecting potassium chloride, it's a standard method, and indeed the heart stops beating, does this flow continue? But beyond that, well, let me start with that. So here's time, and here's blood flow. So at time equals 0, he clamps he stops the heart, and the flow goes way down, but it's not 0, it continues at some value. And then he wants to test the mechanism that we've been talking about, spontaneous flow, to see if that's what's responsible for driving it. And the signature feature of this is that light, especially infrared, is responsible for this. And you can see the result. Infrared causes the flow to increase by about 3 times. And then when the infrared is removed, it goes back down to the control level. So we think from these experiments that indeed radiant energy does help drive blood flow by a mechanism that's the same, essentially, as what we showed in the laboratory, which means that your blood flow is, if our experiments are correct and correctly interpreted, it means your blood flow is driven not only by your heart, but also by this vascular mechanism we call self-driven flow. It's not a great name, but it's driving blood flow through your capillaries. So the question is now, do we still have 10 or 15 minutes?
Oh, no, you have time. No problem.
Okay.
Don't worry about it.
Yeah. Okay. If radiant energy can drive blood flow in your cardiovascular system, in your capillaries, what about elsewhere in your body? Do we use light, radiant energy, for something else besides blood flow? I think the answer is yes. And let me explain how I think it works. So here's a cell. We've got a pretty yellow membrane around the cell. And inside are macromolecules, proteins, nucleic acids, etc., also smaller molecules. And usually these molecules are arranged so the hydrophobic moieties are inside, and the surface exposes the hydrophilic surfaces to the water. And what that does is it builds easy water, you see, like that. So this is what we presume happens inside the cell. But of course inside the cell it's extremely crowded, and even this cartoon is a gross underestimate of the crowdedness. And so basically you have interfacial or easy water all over the place, and it's negatively charged. And you'd expect that if you were to stick an electrode in here, you'd get net negative charge because of that. And it's well known for 60 years that that is the case, although the standard explanation is that it has something to do with the membrane. I wrote a paper 4 years ago suggesting that reasons why that can't be the case, and that a simpler explanation for the well-known negative electrical potential is the water, the easy water that pretty much fills the cell. So that's a side point. But the point is that you've got all these negative charges inside the cell, and they repel each other. They want to get out, and that represents potential energy inside the cell. So this negatively charged easy water is essentially containing potential energy. And this potential energy then gets used in protein folding. Now, what do I mean by protein folding? Well, everything that goes on in the cell is caused by proteins, the work of the cell is caused by proteins, and typically a protein will be in one configuration, say, call it extended configuration, and when it goes to do something, it will fold, bend, and then go back to where it was. So in muscle contraction, for example, if you have a muscle cell, the muscle proteins will do something like that, and then come back when the muscle is relaxed. In a secretory cell, the same thing happens with different proteins as they secrete material. In nerve cells, something similar happens. The proteins will undergo a configurational change to transmit the information along the axon. So that energy is used to drive it. And the way we presume it works, and this is laid out in my 2001 book, is here's a protein right there. It's surrounded by easy water, and what happens is that the easy water melts into ordinary water, and the protein folds. And now, when the, say, muscle contraction ends, you want to go back to the original state, it restores the state, and the easy water builds again, and you're back now to the resting, relaxed state, the stable state. However, if you don't have, on the right side, if you don't have much easy water or no easy water, no is extreme, but basically this is hydration that we're talking about, hydration inside the cell. If you don't have it, then the protein finds itself in a strange situation, unusual, not a physiological situation, and can't restore itself, so it remains contracted, and you might say impotent. What it means is that the muscle cell, if it's a muscle cell, is not functioning well. And if it's not functioning, you have a cramp, for example, in your leg, and all the energy has been depleted. So you need to be able to have enough easy water around to be able to come back to this initial state. And so, if you go through the steps, the potential energy from the easy drives work. Now, what builds the easy? It's light. Remember? Light builds the easy, which builds a negative charge, which gives you potential energy. And the potential energy is used for the work of the cell or the folding of the proteins, connecting the dots. Basically, light input can give you work of the cell or protein folding. So it means, effectively, through these several steps, it means that light that you absorb is able to convert to work that you do to live, to do whatever you do, whether it's sitting on your fanny or running a marathon. So the question is, where do we get our energy? Obviously, we get energy from food because, you get plenty of energy from food. But I'd like to suggest to you, based on what I've been presenting, that you can also get energy from light. The light is absorbed by the water. It builds easy water, which gives us energy. Okay, so it means that we have two pathways for getting energy, and this is the sort of conventional one, although these guys look awfully unconventional. And the other one is similar to what plants do. And some unicellular organisms, they use light to give energy. And I believe the evidence points to the fact that we do the same thing. We actually use light to get energy. How much? Not clear yet. Well, should this matter to you? Water matters, obviously. You need to keep hydrated. Hydration is, from the point of view of inside the cell, it's building easy water. And light matters too. So, that leads to sort of the almost endpoint. So for your health, I think what you'd like to focus on is what can build easy water inside your cells. Because that is certainly one factor to keep you healthy, is to remain well hydrated. That appears in every medical or physiological book, but not necessarily the mechanism that I've been talking about. So the first thing is to drink water, because water is the raw material for building easy water. So when you're thirsty, or even when you're not thirsty, drink a lot of water. Next one is green juicing. I think some of you are familiar with that. What is that? What are we talking about? We're talking about taking a plant, fresh plant, squeezing the juice out of the plant and drinking it. What are we squeezing out? We're squeezing out the water from inside the plant cells. And these are fresh, robust plant cells. And inside the plant cell is full of easy water, the same as should be the case in our cells, but might not be if we have certain pathologies. So by drinking that squeezed water is one way, and some of the gurus of natural health are saying that this green juicing could be the simplest and most effective way of ensuring good health for everyone. Cheap, takes a little effort to do the squeezing, but okay. Another one is various substances have been known throughout the ages, even back in Ayurvedic times, to be good for health. I've listed 2 of them here. We actually studied 7 or 8 different ones that have been known, including turmeric, coconut oil, ghee, and some others. And one of the odd things that struck us is that these substances, even aspirin, these substances are broadly effective. They don't just attack or help one organ or one mechanism. They affect everything around your body. And so the question arose to us, if they're so effective, either they have multiple mechanisms or receptors or something, or there's one mechanism that operates everywhere. And of course we thought of water, because water is everywhere. So we tested these and we published the result, and the result shows that at the concentrations that would be typical inside your body, these all build easy water. So we think that it's possible that these substances, as I said, known for the ages to be good for health, may operate by building easy water in places where you need easy water. Sunshine we don't get so much of in Seattle in the winter time. It's kind of gray and gloomy. When the sun comes out, you see smiles on people's faces. So why are the smiles? Well, it could be a purely psychological effect, or, sunshine, as I mentioned, contains a lot of infrared energy. So we absorb the infrared energy. That infrared energy helps us build easy water. And now the cells that had been deficient in easy water, including your brain cells, suddenly have enough easy water and you feel better, you feel normal, you feel the way you should. So that's a possibility. And the extreme of that is a sauna. So you get in a sauna, take off your clothing, expose yourself to heat. So the heat, it means that you've got a lot of infrared energy around and you absorb that infrared energy. I don't know about you, but I've had some experiences in Finland and Russia with this, and invariably you feel better when you come out than when you went in. And so a simple possibility is the infrared energy is building easy water. Another one is earthing, or we say grounding. The rest of the world says earthing. How many of you know that the earth is not neutral but negatively charged? Okay, not too many, fewer than half. Well, I didn't know it either. It was when a Russian guy working in the lab, just as he was departing back for Russia after 6 months, we started talking about the Earth's electric field. And I said to him, Andre, you're talking about magnetic field, right? No, we're talking about electric field. I said, you're crazy. I studied electrical engineering as an undergraduate, and nobody ever said anything about the Earth being negatively charged. You plug into the receptacle and the third prong goes to zero, goes to ground, it's neutral. He said, no, you have bad education. In Russia, everybody knows, even middle school students know, that the Earth is not neutral, it has a net negative charge. Next morning, one of my students brings me Feynman's lectures, Volume 2, Chapter 9. It has to do with the Earth's electric field and talks about the evidence the Earth is negatively charged and positive charges up there. So I became convinced that that was correct, and I came to realize that this is extremely important. We all think the Earth is neutral. I couldn't believe, I was shocked that I thought the Earth was neutral when the evidence, lots of evidence, but now forgotten, says the Earth is negatively charged. So now, you may know that earthing, connecting yourself like barefoot to the Earth, is good for health. And people have exploited this to devise gadgets and such that you can earth yourself or ground yourself. My son does it. He's working with a computer all day. He has an aluminum plate, and the aluminum plate is connected with a robust copper wire to a rod that's sunk 3 or 4 feet down, and he feels better when he connects himself. Why is that? Well, I think one possible explanation is we know that if you add negative charge to water, you get easy buildup. Easy is negatively charged. See, so it kind of makes sense, and we've published experiments demonstrating that. And so if you walk barefoot on the beach near the water, especially a wet beach, usually you feel good when you do that. And one possibility for why you feel good is you're either getting negative charge seeping into your body and occupying those spaces where you don't have enough easy water and building easy, or getting rid of the positive charges, which are not good for your health. They move toward the negative. So those are some expedients. There are more, but those are a few simple ones that have been known for many years. We don't even think twice about them. I think it's possible that some, or even all of these, could operate through the mechanism of building easy water. I hesitate to say because it sounds a little bit like the guy who his only tool is a hammer and everything looks like a nail. But I think it's possible. So I'm not suggesting that we photosynthesize, but that we use light in a way that's not so different from the way plants use light, especially the first step of photosynthesis, splitting the water into plus and minus. So I don't like to end without practical applications. Just a few slides to quickly show. You might imagine some of the practical applications. And one of them is getting energy from sunlight and water. You know, we can get energy from photocells, right? But we have to deplete the earth of its natural resources in order to do that. We're talking about light and water, renewable resources. And so by now you're familiar with this. This is a hydrophilic surface next to water, easy. And over here we have the proton sitting in the so-called bulk water. And the idea is you put two electrodes in, and these electrodes, if you have enough energy, you ought to be able to light a light bulb and get energy in some way. And we've achieved that and demonstrated it. So what you see here looks complicated, but it's not. Here we have a whole bunch of cells that contain naphthalene and water and pairs of electrodes built in. Here we have a switch, and there's an LED light underneath this magnifier. And so the principle is demonstrated here. Just turn it on, and it lights. And this is from water and ambient light. That's all. So we have a company that we put together to try to make some of these applications, to bring them to humanity, to solve various problems. And we're working on that. Another one is getting drinking water from contaminated water, obviously a serious problem. And we can do it in a simple way. Again, the challenge is to scale it up so that it's really useful. So if you remember, here's a naphthalene tube, right? And you have flow going into the naphthalene tube and through it. And so we, as representative flow, we use water plus microspheres as like bacteria, simulated bacteria or such, and we've studied it. We've also used genuine contaminants, but just showing the principle. So you have an exclusion zone here, and the microspheres are here. We built a collecting device, which is nothing more than two concentric tubes, and the junk gets collected at the center and gets thrown out. And the stuff that's on the side is free of the contaminants, and it gets collected here. And this is a representative result. It's genuinely representative because we can get as much as a 200-to-1 separation in one single pass. So we're excited about this, and we're trying to develop it. It's not so simple, but there are a lot of engineering challenges to make it useful, because the laboratory device, the throughput is such as maybe to satisfy the thirst of a flea, but not you. So we're working on that. And we're also working on the possibility of separating salt from water, because I don't need to tell you about the need for drinking water. The ocean is an obvious place to get it from. And if we can separate the salt from the water, then in a similar way, we have some promising preliminary results. We can take ocean water and create drinking water out of it. You know it can be done now by reverse osmosis, but the amount of energy that's required to do it is horrendous, and only the Saudis can afford it, pretty much. This uses the energy of the sun, ambient energy. So if we can scale this up properly, I think we'll have something important. Okay, so I've taken a lot of your time, and to conclude, the main point is the science, really, and that is that water appears to have 4 phases, not 3. We all know about ice, water, and vapor. We found a 4th phase, and this 4th phase, actually, as I mentioned earlier, the structure of this phase is not so different from ice. It's not ice, but it's not so different. And we found that if you want to freeze water, you must go through this stage to get to ice. And if you thaw the ice, you must go this stage to get to ordinary water. It resolves some of the thermodynamic anomalies that's associated with ice formation. But all of this, I think, has rather broad implications. The main and central point is that radiant energy is getting absorbed by the water. That's kind of the central point. What follows from that is a whole lot of different things. biological applications of that. So I presented evidence that the flow in the cardiovascular system is in part due to flow that comes, it's actually because you get easy build up here and easy build up here. Protons in the middle, the protons want to get out, they go either this way or this way, so you get flow. So, and biological applications pretty much in all cells dealing with protein folding. In chemistry, when you open the chemistry book, an undergraduate textbook, you see a lot of reactions and aqueous solutions and descriptions of the mechanism. If we're right, those mechanisms may be mostly wrong because none of them take into account the effect of light, the formation of EZ, the separation of charge, etc. One would hope that perhaps some of the rather complicated explanations may become simpler if you do take into account the kinds of things that we've observed. The weather. I just read in a magazine article that predictions are getting better and better, but you know, still, you don't know if you should bring in an umbrella to work the next day or not. 50% chance of rain. Well, what do you do? And I think that the reason for the rather poor predictive value, which mostly it's computer models and also information on pressure and temperature. And I think the reason that they're not so predictive has to do with the fact that the most important variable is not pressure, not temperature, but charge. These clouds are charged. And nobody that I've ever seen that gets up on the TV, I don't watch TV, but I know, says anything about charge in the atmosphere, charge in the clouds. And I think if this is properly done, next book, that one can not only predict, but of course understand the genesis of weather in a much more meaningful way. Well, I explained to you why water, especially easy water, is critical for your health. It fills your cells. You gotta have enough of it, otherwise you're not healthy. If you want to process the food by freezing it or evaporating the water and drying it, it's helpful to know something about those processes. And from a more practical point of view, I mentioned to you about filtration, about desalination, and all of which we're working on getting electricity from water. So I would like to say this is my last slide. Actually, there are two more. And one of them, the next one, has nothing at all to do with what I presented, but it may lead into the discussion that we may have afterward. We have an institute that we created. It's called the Institute for Venture Science. And I know some of you know about it. And the idea is to fund promising ideas that may bring scientific revolutions. These are ideas like the ones that are being discussed here, ideas that challenge the mainstream views. We invite pre-proposals from people who have one such or more such idea. And after the pre-proposal stage, we received more than 200 in the last round. We narrowed it down to about 15 that look extremely promising, that if they're right, this is going to change the world. And we invited 5 of them, we invited 15 to write full proposals, and we've selected 5 of these projects, which, if they prove, if their promise really is justified, these are the kinds of approaches that will change the world in major ways, create scientific revolutions. And of course, we're looking for donors because this is not the kind of thing that our government will fund, nor the governments of Europe or Asia or what. So we're looking for private donations. We have some, we don't have nearly enough. And if any of you know somebody who has done well and would like to contribute or return some of this to society in a meaningful way, please get in touch. So I end with the book, this is a book that describes many, though not everything that I presented here, and I think it presents it in a clear way, and many people have commented on the artwork, which is done by my son, who's a professional artist, and new books coming. So thank you very much for listening. Much appreciated.
Okay, we're gonna have questions now. Those who want to have a question, please get a microphone and queue yourself up.
Yes, I was looking at an application for filtering water. The Engineers Without Borders were going to remote areas and building, taking 55-gallon drums and filling them up with alternating layers of gravel and sand and straw, etc. And the operational characteristic of this filter required that a gel or film called schmooze deca, which is kind of a microbial aggregation of material at the first layer, was required for the filter to work to filter out all the contaminants and bacteria. And I was wondering if it might be that schmooze deca is like this Nafion, and it's actually creating this easy water layer, which then goes through, and none of the small particles, micron-sized bacteria or any other contaminants get through that. Schmooze deca is in a similar way to that it may be a porous Nafion. Any comments on that?
Schmooze deca, it sounds like a new creation in the latest Japanese restaurant.
Yeah, I think it's a German word.
Oh, okay. It's possible because we've done experiments that follow roughly along those lines. So we take some filter paper and we pour the water that goes through the filter paper, and the stuff that comes through, of course, is filtered as you'd expect, but also it contains easy water. And we think it's that layer of cellulose. We know that cellulose builds EZ. Cellulose that lines all the pores of the filter, and some of that comes through. And so not only should this device filter, but also the water that comes through will be good drinking water because it contains EZ water. So I think yes, maybe you're right in your supposition about what happens.
Yeah, maybe some graduate student could study it's a biological film, study the properties of that and help or use it in your process, in your institute.
Yeah, could be, sure, yeah. By the way, the institute, you mean the Institute for Venture Science that I mentioned, yeah, this is a bottom-up institute, it's not a top-down, so we don't dictate we invite anybody and everybody to submit their proposals, and from that we choose. So that's the way it works. So if one of you wants to do that and make a submission, right now submissions are closed because we want to be able to fund the people who we've selected, but it will open again.
Are there other liquids besides water that could possibly exhibit the same type of behavior?
Yes. To my dismay and surprise, I thought water was unique. I regard myself as a kind of biologist, and every biologist knows that there's no life without water. Nothing happens without water. So there must be something unique about water. And I was really disappointed to find that other substances create EZs. For example, ethanol. You could put ethanol next to a Nafion and you'll find an EZ next to that. Also, other small polar molecules, DMSO, for example, and a few others we found. We published a paper about 10 years ago demonstrating that it's not just water, but other substances. So I don't know, it raises a kind of, this is a philosophical organization, Chappell Natural Philosophy, and so it's possible, if easy water is a necessary condition for life, and I absolutely think it is, there might be the equivalent of EZ that you can get with ethanol or with some of these other solvents, and that would extrapolate to a possibility that some kind of ethanol-based life could exist somewhere. I mean, it's a huge speculation, and I don't stand by it, but I'm just suggesting that something like that could conceivably be possible, which is very disappointing for someone who believes in water so much. But thanks for your question.
Very interesting talk.
Thank you.
I was really taken by this. So, would there be a possibility of generating electricity by shining, say, sunlight onto water?
Absolutely.
With some kind of
Absolutely.
negative terminals?
Absolutely. Yeah, I mean, it follows directly from what we presented. The problem is a technological one. It's not a theoretical one. If you want to generate electricity, you need something big, right? If you want to do it for a home or a town or something like this, you have to scale it up. And we found, other people knew, but I'm learning that the scale up itself is a formidable problem. It's not so simple to do it, and it needs investment, especially. You need to have the people who are building the devices, testing them and whatever. And I think this is quite possible, but it depends on adequate investment to build prototype devices, to tweak them and make sure they work properly. But I think there is something in this for the future, definitely.
Certainly buy this book and get my students to do 1 or 2 experiments.
Well, thank you.
Further on to that.
If you buy the book, I'll become rich.
Thank you very much. No, well, thank you.
Okay.
So I want to ask you about some other things. So does this have anything to do with like the homeopathy where they say they can impress the molecule of whatever they're trying to treat you with, such that there's none there, but the water still retains those properties? Have you looked into that sort of thing?
Well, we're not experts on homeopathy. However, I was invited to present at a conference in London last summer dealing with homeopathy. It was a special conference because it was held at the House of Lords and the Royal Society of Medicine. So it was a big deal. And I was one of 3 or 4 major speakers. And our contribution was information in water, because homeopathy suggests that there's some way to store information in water. But what impressed me most was the contribution from some Russians. And I'll tell you about it because it's pretty interesting. And the person who did the studies was not there. It was presented by another Russian, a friend of his, because the original guy was ill. The fellow who did the studies, his name is Alexander Konovalov. And he's not just your average sort of scientist. He was head of a major chemical institute in Kazan in Russia, and he also then became the rector of the university for 11 years. And this is the university that ranks just behind the top Moscow University, St. Petersburg, Kazan, and he was the rector. So he's a full member of academician, which is in Russia the highest level. He's a formidable guy, and he became interested in what happens when you do the dilutions that are associated with homeopathy. So I don't know if everybody is aware. You take some substance and you dissolve it in either water or alcohol, and then you take 1 part of that, 9 parts water, or 99, and you shake it. They call it succussion. And then after that, you dilute it again, and you dilute it again, and dilute it again, go through the same process. And when you do it enough times, statistically speaking, there are no more molecules left of the original. There's only water that had been in contact with the original substance. And if water has no information, no memory or whatever, you'd expect nothing. You can't get any effect. But clinically speaking, many people have shown a huge effect of homeopathy. In fact, it's kind of rivaling the pharmaceutical companies who don't want homeopathy because they can't make any money from it. So homeopathic people have fallen on hard times and they're under attack right now. So Konovalov found something incredible. And I give his background to start with because this is a top-level scientist. So he takes the water and he dilutes it, and you get pretty much what you expect. The substance is diluted, and shake it, and you do it again. Everything is fine for the first 3 or 4 dilutions. When you come to the 5th or 6th, something unexpected happens. You begin to pick up what he calls nano aggregates in the water. And these are, he knows the size. He doesn't know what's in these aggregates, but the only thing that had been present are the original substance and water. So it's some kind of combination of some special water, could be easy water, something, and the original substance. And then he dilutes it again, and these things get bigger, and you get more of them. And he dilutes it again, and they get bigger, and you get more of them. And he dilutes it again, and they get smaller and fewer and smaller, and you go down to the baseline. And 4 or 5 dilutions later, the whole thing repeats again, an infinite number of times. So what it means is that when you dilute, you don't get what every chemist would tell you you get. You get something different. And it may be that these differences are what is responsible for the homeopathic effects that are observed. He said, if you don't shake it, you don't get this effect at all. You have to shake it, the succussion I mentioned. And if you isolate it from using new metal and such from the environment, it doesn't work either. You have to have the environment giving energy, and you have to undergo this shaking. Those two are critical factors. That's what the homeopaths do. So I'm sorry I gave a long answer to a short question, but the science underlying homeopathy is different from, there are certain findings that support the possibility that something happens when you go through these dilutions and shaking that's completely different from what every chemist would expect. There are lots of papers from this guy translated into English. His name is Konovalov. I highly recommend it. So this is very important for the whole field of homeopathy, which tends to be discredited because, after all, how can it work if you dilute it to the point where there's only water? The water would need to have some information, some memory, and also the findings of Konovalov that I just mentioned are highly relevant to that. So it's a different, after this symposium, quite a few people came away with a different view of homeopathy. Does that answer your question?
I've also seen some things in the back of the magazines about energy water or triple distilled water or something like that. Have you seen those advertisements, and are those scams?
Well, we don't know which ones are scams and which ones are not scams. So energy water, what I present that suggests that water can contain energy. So the idea of energy water, for me, is not outrageous at all. Whether what they sell has that's another story. I have no idea if it does or not. There are hundreds, at least more than 100 companies that sell various kinds of water. People are becoming tired of the junk that's in our tap water, and so a lot of companies have sprung up to produce waters that should be cleaner and better for health. Whether they are or not, I'm not sure. We did look at 1 or 2 of those way early on, but we don't test waters in general, because people come to us and they say, which water should I drink for my health? And I'm not sure which one. And we don't like to test the waters because then it appears that we're endorsing a particular company. We don't want to do that because we're a scientific group, we're not a commercial group. And so we stay away from that sort of thing. And the downside of that is that having not done the test, we don't know which waters contain a lot of easy water, for example, which ones have energy, which ones don't. Someone should do it. It really needs to be done, because this is such a widespread question that everybody wants to be healthy, and people have come to know that water is really important for your health. And the question is always, which water? If the NIH would spend $5 or $10 million supporting a study to test all the different kinds of waters that you can purchase right now, I can't imagine a better investment. It takes it takes that much money to carry out studies like this because to hire the people, to do the experiments, to get the animals or people, it's a big job. It's not a trivial job. And at one point we were willing to do that and put in an application to the National Institutes of Health. And it seemed to me that they never heard the word water. That water is something that is not part of health. Water is water, you know. If you want to make a big deal of water, there's something wrong with you because everyone knows that water is simple. So after a while I realized that this was not going to make it, and so we gave up. Someone to do it, some person who's independent of any of the companies producing the water, and someone who knows something about water. Needs money.
Yeah, have you done any absorption spectroscopy?
Yeah.
Is there a difference between regular water and easy water?
Yeah, so one of the things, we focused, especially lately, most on the UV vis portion of the spectrum, because it tells us something about energy. And we found, first of all, if you look at that region of the spectrum, easy water absorbs light at 270 nanometers. So if you put it through the put it in a cuvette and a spectrometer, you see an absorption. This is wavelength, and this is absorption. It looks like this with a peak at 270 nanometers, and that's how we know. That's how we know if a sample contains easy water. The higher the peak, the more the easy water. And there are other effects too in looking at the UV vis spectrum. Magnets have an effect. We found that recently, and we're trying to figure out what's going on. So yeah, we've done that, and with interesting results. You had a question?
Yes, I've watched a YouTube video
Yeah.
showing engine that runs from water instead of gas.
Yeah.
What do you think about that?
I think we have someone who has expertise here in that area, but all I can say is that I've seen enough in terms of demonstrations showing that to think there's something going on. And what makes total sense to me is that we know that the water molecules are split into positive and negative by light, and when they recombine, you get energy release. And so this is conceivably a powerful mechanism to create potential energy that can then be delivered. I know that various cars are reputed to have run on water instead of gas. In Japan, at a company about a year ago, I had the pleasure of riding in a car that was fueled partly by diesel and partly by water. It was a company that's producing a special kind of water, and they actually demonstrated to me a diesel engine, and had it set up in such a way that you can see the output of the diesel engine as a kind of fire that comes out into a room with asbestos shielding. And you have an infrared camera looking at the fire. So first they start by putting diesel fuel in the engine, and you get a big bright fire, and you can measure the temperature of the fire using the infrared camera. Then they mix it 50% diesel and 50% of their special water. And you get the same fire, but it's brighter, and it's higher temperature by something like 200 degrees. So my own experience with this sort of thing is that it looks like it's real. And there's someone who'll be coming talking about Brown's gas, which may be similar to the kinds of things I was talking about, not sure. At our water conference, we hold each year, this is the 14th edition, conferences on water that discuss many of the kinds of things that we've discussed here. This year it's in Germany, near Frankfurt Airport, just a 20-minute car ride. If anybody's interested, it's waterconf.org. You can find the program and such there. It's an exciting meeting, very informal, very friendly, and some of the latest and most interesting developments in water. Thank you.
Thank you.
Yeah. Please.
Do you think easy water could play a role in the development of the embryo?
In what?
The development of the embryo?
Sure.
Absolutely. Differentiation.
Yeah. So, yeah, and it's because it's directional. So one of the lingering questions is how does the embryo know which way is up, which way is down, et cetera, et cetera. You need something that is polarized. And the embryos are mostly water, more than we are. Embryos are 90%, 95%, I don't know what the number is exactly, but the easy water has a directionality to it. It grows from one point and keeps growing out. So it's possible that that water can confer a kind of polarity or directionality on the growing embryo. It could be very important in that field. I don't think anybody has studied that yet. But thanks, that's a great question.
Any more questions? Well, if not, thank you very much for the talk, and then we'll have a break and then come back and do some more talking with Dr. Gerald Pollack. Thank you.
Thank you.